You should be able to see the quiz. Tell me if you can't. And I did send you this document, the quiz with the answers, earlier today. So you have that for study purposes and to compare your answers with the grading. And I want to emphasize, if you have any issues with how your quiz was graded, let me know. Okay, the first question, what were Mendel's...
Professor?
Yes?
Could you zoom in, please?
Is that better, or you want bigger than that? I can make it one bigger. Okay, hang on. How's that? You see that better?
It's better.
Okay.
Yes.
All right. What Mendel did was before he started doing crosses, he developed pure true breeding lines by inbreeding, doing one selfing generation after another until all the loci were homozygous for each of the traits. So that was one thing that no one else had done before him. And the other is, after doing a cross and getting a progeny, he mated those progeny with each other so that he could go to an F2 generation, and that's where the idea of dominance came from. Now, some of you confused question 1 with question 4, Mendel's laws of inheritance. So you gave me Mendel's laws of inheritance for question 1, which is not correct. Mendel's laws of inheritance would not have been possible without his 2 innovations that were called for in question 1. So is that clear, the distinction between Mendel's laws and the innovation that allowed him to arrive at those laws? Okay, question 2, most of you got this right. The cross is the mating of 2 individuals. An F1 is the progeny of a cross. Inbred results from multiple generations of inbreeding, homozygous, identical alleles at the same position. Hybrid vigor, many of you did not get this. Hybrid vigor is the result, or is progeny that are more robust in one way or another, or multiple ways, but more robust than either parent. And the F2 generation is progeny of a cross between 2 F1s. Then number 3, and stop me if there's any, if you have any questions as we go. Distinguish between phenotype and genotype. Most of you got this right. Phenotype is outward appearance. Genotype is the genetic makeup represented by symbols. Then we come to Mendel's laws. We had to pick them out from the list: segregation, dominance, and independent assortment. And then number 5, again, most of you got this right. The discovery in the 1970s of reverse transcriptase completely revolutionized molecular biology, because now you can go backward from RNA to DNA. The polymerase chain reaction, I got lots of different answers for this, and I gave either full or partial credit to most of them. But what I was looking for here is that the real merit of PCR is that you can produce abundant DNA, in principle, as much as you want, from very little target material, as little as a single molecule. Then number 7, I gave you a question about plant flower color. And I said, how would you determine if flower color is controlled by a single locus? And if it is, how would you know which trait is dominant? Well, again, I got lots of different kinds of answers to this. And I gave credit for where I thought you were on the right track. Some of you got it completely right. But you would cross the plants with the 2 different flower colors. If all the F1s are the same color, you've identified a single locus. And the color of the F1 tells you which trait is dominant. So if all the flowers are white in the F1, then white is dominant. Then number 8, indicate which of the following statements is correct. They're in red. Homologous chromosomes have identical alleles at all or at most loci. The next 2 statements are incorrect. A series of inbreeding generations will produce a true breeding line. That's correct. A locus designates a particular physical position on a chromosome. And alleles are not identical forms of a gene, they're alternate forms of a gene. And finally, a trait that disappears in the F1 and reappears in the F2 is recessive. And number 9, this is a question that Scott posed. And he wants to know with this animal called a fuzzle. Number 1, if in the F1, if you cross black and white fuzzles, in the F1, if they all have white coats, what is that? Choose from the list below. It's complete dominance. If they have gray coats, it's incomplete dominance. And if they have patches of black and white fur in the same animal, that would be codominance. And then, Scott, would you like to discuss the last question, which is one that you posed?
Sure. This is a question on obviously sex-linked inheritance. The question gives us a female carrier, which means she is a heterozygote. One of her X chromosomes is dominant and one of her X chromosomes is recessive. And she is having children with a man who lacks the same disorder, which means his X chromosome is dominant and he has a Y chromosome to complete his set. So that's the setup. And the question asks or states it's possible for a son to have the disorder but not a daughter, and it asks you to explain why. So if we set up a Punnett square using the X, and in this answer here, the asterisk is indicating the recessive chromosome. So we have X star crossed with a man who is XY. And if you put this out, you would see that the daughter, she gets an X from each of her parents. That would be shown in the Punnett square. She gets an X from each of her parents. One of the daughters would get the dominant from the father as well as a dominant from the mother. A second daughter would get the dominant X from the father and a recessive X from her mother, which would also make her a carrier, but she would not have the disorder. The other daughter would get dominant X from both parent and be completely unaffected. With a son, however, the son gets the Y chromosome from his father, and the X chromosome comes from his mother. So one of the X chromosomes would be dominant, and he would be normal or wild type, you might hear it called, and the other would be the recessive X, and he would have the symptoms or express the disorder. The reason being he has no second X chromosome, a dominant chromosome that might mask the disease. So the problem with X-linked chromosomal disorders is it tends to affect men more than women, simply because men don't have a second X chromosome to mask these things. Color blindness would be an example. Some of the male pattern baldness can come in. So there are, plenty of real world examples of this. That would be pretty much a summation of what a good answer would be.
Okay, thanks, Scott. Any questions for Scott about the reasoning behind this answer? Okay, as I said, you have this document with the answers. I sent it as an announcement so you can review it and compare your answers, the answers that you gave on the quiz, to these answers. Okay, if there are no more questions, I'm going to stop sharing this screen. And share this one. You should see a slide that says natural selection and evolution. Tell me if you don't see it. Okay, natural selection and evolution. They determined life forms since the beginning of life. They continue to do so. So what are they by definition? Well, natural selection is the differential survival and reproduction of individuals due to differences in phenotype. It's the key, natural selection is the key mechanism of evolution. What does natural selection need in order to operate? Well, two things. First is variation. And you know all about this. We've discussed it several times. The sources of variation are mutation, recombination of genes at prophase 1 of meiosis, and random assortment of chromosomes at metaphase 1 and telophase 2. So make sure that you can make this connection that natural selection, in order to operate, it needs variation, and it comes from these three sources primarily. The other thing that's required is overproduction. That is, there has to be an excess population with variation in that population so that natural selection has something to select from, can choose out the best from a number of possibilities. Is that understandable? Okay, then what is evolution? Well, evolution is an inherited change, and I've italicized inherited because that's a critical distinction from other types of changes, which we'll discuss later. But it's an inherited change in a population over successive generations, leading to the appearance of new forms. Now, the environment on this planet is constantly changing over time. And as it changes, it creates new niches. And these new niches then are occupied by life forms that are specifically adapted to them. So as new opportunities arise, natural selection will fill in those opportunities. Darwin's theory. Well, Darwin lived from 1809 to 1882, and he published his seminal work in 1859. But this was after a long period, roughly 30 years, of studying and travels. And in 1859, he published this book, The Origin of Species by Means of Natural Selection. And that started, took a long time, but it started a scientific revolution, showed that life on Earth is constantly changing and only the fittest organisms survive. If you want to watch a video, a description of evolution, there's a URL up here on the right that you can put into your browser and get a very simplified description of what evolution is. It's about 8 minutes long. Okay, Darwin studied for about 30 years before he published, but the most important period during that 30 years was a 5-year voyage around the world that he took, starting in 1831 and ending in 1836. And the most important part of that voyage was a stop at the Galapagos Islands off the coast of Ecuador in 1835. And on the right here, you see the path of Darwin's trip. He started in Plymouth, England, went south to the coast of South America, the East Coast, made several stops there, went through the Falkland Islands, Tierra del Fuego, then up the west coast and ended up in the Galapagos Islands, which we'll talk more about. And the important thing I want you to know is where the Galapagos Islands are. Off the coast of Ecuador, and it's a whole collection of small islands on which Darwin noticed that unique life forms had developed over a long period of time. So this was one of the places he stopped that gave him great insight. Here's a drawing of a picture of the ship that he was on. And it turns out Galapagos in Spanish means turtles. And the reason these islands were named Galapagos is because of these giant turtles. Here you can see how big they are compared to a human. In fact, Darwin claims that he had climbed on one and rode it at one point. But these animals, they still exist, but they almost became extinct during the whaling, the height of the whaling industry, because they're a convenient source of meat. You can put a turtle on a ship and sail for months without ever feeding the turtle anything, but you can harvest them one by one to feed the crew. Fortunately, they didn't all have that same fate. Now, Darwin's studies were secondary. The Beagle didn't go around the world for him. He went around the world because that's where the Beagle was going anyway. The ship's captain had his goals, his priorities. What were they? Well, first of all, the British government wanted him to survey the coast of South America and secure the safety of British shipping. Secondly, I don't know why this was so important, but Fitzroy wanted to return 3 natives from Britain to Tierra del Fuego, which is clear at the southern tip of South America. And Darwin had made a quote in one of his writings that to settle these natives in their own country was one of the chief inducements to Captain Fitzroy. Now, a third goal was religious. The Anglican Church, in the person of a Reverend Matthews, tried to establish a mission to the Fuegans. And that effort ultimately failed. And finally, at each stop, so the ship was stopping frequently along the coast, and at each stop while the crew was surveying, Darwin would go on shore and record observations and collect specimens. He collected thousands of specimens of both plants and animals and sent them to England for study later. So that's the real purpose of the Beagle. It turns out that Darwin's studies, even though they weren't a top priority, turned out to be much more important than anything else that the Beagle did. So this is sort of a simplistic pictorial of the evolution of finches in the Galapagos. So if we start the upper left, 2 million years ago on the Galapagos lived birds named finches. At first there were only 2, but they mated and made a large population of finches. And as the population grew, variation crept in. So variation in size, variation in color, and in other characteristics. Eventually, individuals or groups with different characteristics began to mate with each other, and these variations included different beak sizes, different feather colors, sizes and shapes, and many other things. And as time went on, some of the variations were unfit and they died. And what eventually happened was that individual or groups of different types collected on different islands in the Galapagos. And this is what Darwin noticed on his voyage. So one of the main things that Darwin looked at was the adaptation of finch beaks according to the food supply that they had. If you look in the upper right, you can see that some of the finches, and these are correlated with islands that they lived on. Some of them ate seeds and lived on the ground. Some ate cactus. Some ate insects. These lived in trees. And some ate fruit. And what Darwin noticed is that the size and shape of beaks was absolutely correlated with what the bird ate. So the ones that ate seeds have these big, heavy beaks that gave them strength to crunch the seeds. And it turned out that the ones with the biggest beaks ate the biggest seeds, and with the smallest among the ground inhabiting finches, the smallest ones ate the smallest seeds. And on the upper left here, you see an elaboration of that. On the lower right is an original drawing that Darwin did showing the various different beak types that he studied. And just out of curiosity, I put another drawing of Darwin over on the left of the turtles. He was quite taken with the turtles. And his quote is, in red here, "It is a circumstance that several of the islands possess their own species of tortoise. It strikes me with wonder." Now, here's where Mendel comes in. Darwin made these observations, and they were critical. But Mendel provided a genetic explanation for this. And I've listed here in yellow 3 genes that have been shown to be important in the variation of finch beaks. One is BMP4, it's called for bone morphogenetic protein 4. It's active in embryo development, and the differential expression of this gene results in variation of beak size and shape. So one gene alone can account for some of the variation. Another CAM calmodulin, modulating protein gene, regulates many pathways, including the pathway for the growth of beaks. And ALX, remove this, ALX1 encodes a transcription factor affecting the face, the development of the front of the head. Variations in ALX1 are highly associated with beak shape and diversity. AXL1 is a transcription factor, doesn't actually code for a protein, for an enzyme, but a transcription factor is a protein that binds to a gene to control whether it's on or off, and if it's on, what its rate of transcription is. So this is all about beaks. Darwin studied many things, but one of the most important in terms of his thinking about evolution had to do with the beaks of finches in the Galapagos. Okay, natural selection evident in just 2 years. We're still on the Galapagos, and we're looking at 2 years, 1976 and 1978. The year in between, 1977, there was a drought in the Galapagos. And that drought reduced the number of small seeds available to finches. And you remember that among the ground-dwelling finches, some had large beaks and ate big seeds, some had small beaks and ate little seeds. And so during this drought of 1977, the number of small seeds was reduced, and many of the small-beaked finches starved. Their beaks were too small to eat the big seeds, which was all that was available. Now, this caused an increase in the finch's average beak size between 1976 and 1978, and that's what the two graphs show. In 1976, the mean beak depth, you can see what depth means here, was about 9.5 millimeters. 2 years later, the mean beak depth was a little over 10 millimeters, and statistically significant. So the reason for that is that birds with bigger beaks were selected over those that had small beaks, because the ones with big beaks could eat the food that was available, which was the big seeds. So is that clear? What happened here, and why the population changed, and why it was a genetic change? When Darwin first published his theory of evolution in 1859, there was a lot of skepticism, including in the scientific community. And biologists in particular didn't really accept it. And there were a number of reasons why. But I've indicated a quote here representing scientific thought of the 1800s. The quote is, "Innovations cannot possibly come into existence through accumulation of many small steps. And even if they can, natural selection can't accomplish it because incipient and intermediate stages are not advantageous. Now, that was the contemporary thinking in the time of Darwin. We know now that this is not true, that evolution can proceed in small steps, and it almost always does, and that natural selection can produce the small steps that are needed for evolution to occur. But by and large, people didn't take, especially biologists, didn't take Darwin's theory seriously until the turning point was the peppered moth, which lives in England, where Darwin and many other biologists were. But the observations of the peppered moth convinced most biologists that the evolution theory is valid. Okay, what's the peppered moth? Well, there are two versions that are relevant to us, a light-colored and a dark-colored. Light-colored peppered moths are better camouflaged against a pristine environment. And it's important to note that the predators of these moths are birds. So birds will eat them if they can see them. So dark peppered moths are better camouflaged against a sooty environment. Thus, as the Industrial Revolution progressed in 19th century England, the color of the moth population shifted from light to dark. So the Industrial Revolution caused tree trunks to darken from soot. Dark peppered moths were better camouflaged on these dark tree trunks than the lighter colored ones. And the dark colored moths population increased as the light colored ones decreased. And here you see it illustrated. We start on the left. You can see a light colored moth on a leaf. And below that, a light colored and a dark colored moth on a light tree trunk. And below that, an even more dramatic example of a light tree trunk. There are two moths there, and they're very difficult to see. So if you're a bird and you're hungry, you probably wouldn't notice those moths on a light tree trunk. Well, conversely, on a dark tree trunk, you see from the polluted environment in Manchester, you see a light colored moth and a dark colored moth. So now the light colored moth is much easier to see than the dark one. So if you're a hungry bird, you will go for the light colored moth. And the picture below that shows the same thing. On a city tree trunk, you can hardly see the dark moth at all. Now, so while the Industrial Revolution was progressing, it only progressed in certain parts of England. So I'm contrasting here two different locations. Dorset, which is on the southern coast. It's primarily a fishing area with tourism, and there's no industry. And so its environment is clean. Manchester, which you can see way up here, is in the center of England. Its economy is heavy industry, and in the 1800s even more so than it is now. So there was a lot of air pollution, which caused tree trunks to turn dark. So that explains why the population of peppered moths in Dorset was largely white, light colored, whereas those in Manchester was largely dark colored. And this graph here shows the result of an experiment where an area was was identified, and a mixed population of moths was released in this area where all the trees had light-colored bark. So at the beginning of the experiment, at time or moth generation 0, there were 50% light-colored moths, 50% dark. And then the moths went through breeding generations. After one generation, the population was still about 50-50. But after 2, now the birds were starting to prey on the dark moths, because remember the trees were light colored, so the dark moths got eaten. And as generations went on, the light moths, again, were better camouflaged, and their population increased while the dark moths decreased. So you can see over just a few generations of moths, the effect of selection pressure. Any questions about the peppered moth and why these observations convinced most biologists, even at that time, that there was something to the theory of evolution? Okay, so we've been talking about the macroscopic level. You can also watch evolution on a microscopic level. So for example, with bacteria, natural selection and evolution in a test tube. So at the top here, you see a population of dividing bacteria, and they're all green. Below that, during division, one bacterial cell undergoes a mutation in its DNA that results in antibiotic resistance. In the third level down, a new environment is created. And that's done by adding an antibiotic to the medium, to the environment. So now we have a new environment that the green population of bacteria are not adapted to, but the mutant is. So you end up with all the green bacteria disappearing, and there's left just a red one. And as it divides, it creates a whole new population which occupies this new niche, this new environmental niche that is created by the addition of the antibiotic. And again, this is a true genetic change. It's caused by a mutation to antibiotic resistance. So my point here is that you can see natural selection and evolution occurring in populations that are macro populations and in micro populations. It's occurring all around us all the time. Now, the point I want to make here is that there are changes that do not reflect evolution, one of which is adaptation. So make a note of this. Make a note of the distinction between adaptation and evolution. Adaptation is not the same. Adaptation is a component of the evolutionary process. The difference is that it is not heritable. In adaptation, individuals adjust, whereas in evolution, populations adapt. So in adaptation, there are short-term changes to suit the habitat and the environment, but they're not permanent, they're not genetic. In evolution, it's a long-term process wherein changes occur at the genetic level for better functioning and survival. And I've got two examples here of adaptation. One on the left is the Arctic fox. In the summer, the Arctic fox is dark colored so that it matches the brown dirt and where it lives. In the winter, Arctic fox is white so that it matches the snow. In both cases, what it's trying to do is camouflage itself against predators. And similarly, on the right, the Arctic ptarmigan, it's a type of partridge. In the summer, it's brown with dark stripes, so it's camouflaged. In the winter, it's white. Now, these are adaptations. This is not evolution, because as time goes by, the ability of a population or of individuals in a population to switch colors just continues. So the ability to switch colors is genetic, but it's the switching of colors is not. So over time, the population never changes. They always have this genetic ability to switch with the season. Okay, is that clear? The difference between evolution and adaptation. Okay. Now here's one that I put in just because I think it's cute. Chameleons. Because it's another example of adaptation, and it's not, the color changes are not evolutionary changes. So how do chameleons change their skin color? Well, their skin is underlaid with crystals of guanine. Remember guanine? DNA. But in the chameleon skin, it's concentrated into crystals. And to change color, the spaces between the crystals change so that the wavelength of light reflected off the crystals changes and the color of the skin changes. And you see examples of how dramatic these color changes can be. Again, this is an adaptation. It's not evolution. Now, what are the functions of color change? It's most commonly thought that it's camouflage, but not really. Most chameleons use color change for social signaling, either to attract a mate or to repel a foe. And it's also it can be a reaction to temperature change. Occasionally, camouflage does work to avoid predators like birds and snakes. The color change can avoid the vision spectrum of the predator. But that's a minor use of a color change in chameleons. Mostly it's social signaling. So again, the point here is that this is a type of change, but it's an adaptation. It's not evolution. And now, to finish, I'd like to talk about evolution of us. Where did we come from? How did we get here? And where are we going? This is a map of the world. Humans came from Africa. Humans, Homo sapiens, evolved just once in Africa, along with other species of Homo, including Homo neanderthalensis, the Neanderthals. And what you see here in red is where Homo sapiens came from. They migrated out of Africa through the Middle East, and then populated Northern Europe, and finally migrated to the entire world. The Neanderthals, shown here in a different color, also migrated out of Africa And you can see the range where they occupied here in Northern Europe. So we're going to talk now just about Homo sapiens and Homo neanderthalensis. The Neanderthals left Africa about 400,000 years ago. 400,000. And they had a tough life. They eked out a living because this was during the ice ages, several of them, and glaciers in Europe and Central Asia. And these poor people from Africa, not people, poor Neanderthals, were trying to survive. And in doing so, they acquired mutations for adaptation to the harsh environment. So they evolved. They existed in just a few isolated communities in Europe, Middle East, and Central Asia. Now contrast that to Homo sapiens, humans. Modern humans left Africa about 60,000 years ago. So that's 340,000 years after Neanderthals did it. So that's a big time gap. Now, the African climate had been warm in equatorial Africa, tropical home for Homo sapiens, and they were unprepared for the harsh environment of Northern Europe, just as the Neanderthals had been. So Homo sapiens that migrated out 60,000 years ago had a choice. A, they can spend, excuse me, the next 400,000 years evolving to fit the climate as the Neanderthals had done, or B, they could mate with the Neanderthals and thus borrow their genes for adaptation to the European climate. And so what did they choose to do? They did B. They mated. So any questions about the map or the migrations or the time periods? So how do we know so much about the Neanderthals and humans? Primarily one man named Svante Pääbo. He's at the Max Planck Institute for Evolutionary Anthropology in Leipzig. In 2010, he sequenced the genomes of 3 Neanderthals. And the genomes were obtained from bone fragments preserved in a cool, dry cave in Croatia. Now this is astounding because the first human genome, Homo sapiens genome, sequence was completed in 2003. 7 years later, Pääbo had sequenced 3 Neanderthals. How did he do it? You all know the answer. How did Pääbo get DNA from Neanderthals? It's right here. PCR. Without PCR, would not have been possible. So he could have as little as 1 molecule of Neanderthal DNA and then make a pound of it if he needed that much. So technology is critical for our advance in knowledge. This is a picture of Svante Pääbo. Here's a picture of a red-haired Neanderthal woman. Imagine from the DNA and bones found in one of the Croatian caves. By analyzing the DNA sequence, it could be determined that she was inbred. Her father and mother were half-siblings. Now at the bottom here, I have the percentages of Neanderthal DNA in contemporary humans. Africans have none. And you can see why. Remember from the map, Neanderthals left Africa long before humans did, and so if they made it in Africa, there are no markers to indicate that. But there are markers to indicate that mating did occur in Europe between humans and Neanderthals. So that now Europeans and Japanese have about 2.7% Neanderthal DNA, Chinese about 2.5%, Polynesian 4%, and me 3.1%. I had my genome sequenced, the exons sequenced. And so I'm sort of high in Neanderthal DNA. I'm in the 97th percentile. And any of you who have had your genome sequenced, you can look and see what percent Neanderthal you are. Now, I'd like you to think about how this happened. We don't know the social circumstances under which humans and Neanderthals mated. All we know is that they did. So regardless of how it happened, there was a parental cross. You see at the top here. So that in the genomes of the offspring, so a child of a cross between a human and a Neanderthal had 50% human DNA and 50% Neanderthal DNA. Now, such a child, when he grew up, he or she, could have mated with another Neanderthal, or could have mated with a human. Now, those who chose to mate with Neanderthals, and if they continued to do that, became extinct along with the Neanderthals. However, those that mated with humans and continued to backcross to humans became us, where we are today. So after backcross 1 between an F1 and a backcross to a human parent, now the genome of that offspring is 75% human, 25% Neanderthal. And after the second backcross, 87% human. After the third, 93%. And after the fourth, 96%, and the fifth, 98%. Which means that after 5 backcross generations, between 1 and 3% of the DNA would be Neanderthal. And that's the range we have today. If you remember, my genome, for example, is 3.1% Neanderthal DNA. So that after only 4 or 5 generations, the percent Neanderthal DNA in the human genome stabilized at between 1 and 4%. And this it could have taken as little as 80 to 100 years at 20 years per generation. And that's where it stabilized. So what that tells us, the conclusion is that it is this proportion of the Neanderthal DNA, Neanderthal genome, that's important to humans. And the reason we know it's important is that if it were not, it would have been lost over the hundreds of thousands of years of natural selection on the human genome. So there's something about this 1 to 4% of Neanderthal DNA that humans are finding useful. Again, if it were not useful, natural selection would have gotten rid of it. Any questions about the genetics here, the proportion of DNA, and why backcrossing to Neanderthals didn't work and backcrossing to humans did?
I have a question. Why do you say it's useful and important?
Because if it weren't useful, if we weren't using it, natural selection would have eliminated it. Remember, that's the basis of natural selection, that if it's important to occupy the niche, it's selected, it survives. It's not important, becomes extinct.
Thank you.
So what are the genes from Neanderthals that are essential to humans? Well, we know some of them. So here are some of the phenotypes. Upper left, fair skin and freckles. Fair skin is an advantage at northern latitudes because it is efficient at generating vitamin D from weak sunlight. Freckles are clusters of cells that overproduce melanin, and they are triggered by exposure to sunlight. So light skin and freckles are remnants of Neanderthal characteristics. There's a gene called BNC2. It's a Neanderthal gene. It's involved in skin color and freckling. 70% of Europeans have at least one allele of the Neanderthal gene for BNC2. Hair. Africans, they came out of Africa, had bushy, kinky hair. Most Europeans now have straight and thicker hair. Humans have Neanderthal genes for production of keratin, which results in the straightening and thickening of hair, which is insulating and would have conferred an advantage in the cold northern climates. Another phenotype is big eyes. Neanderthals required large eyes to enable clear vision in weak sunlight. And here you see a picture of a model from the Ukraine with her very large eyes. So these are just examples. Much of the 1 to 4% of Neanderthal DNA has no known function. It's there for a reason. If it didn't have a reason, it wouldn't be there. And there are only a few of the genes that we know about that actually have phenotypes in humans. Now, some Neanderthal traits persist in humans for no obvious reason. So you see here on the upper left, a Neanderthal skull and a modern skull. The distinguishing features here, Neanderthal skull has this heavy brow ridge and also has this protrusion at the back of the skull called an occipital bun. Human skull doesn't have either of those. And yet you can see those phenotypes in populations today. Here's an individual with a heavy brow ridge, like a Neanderthal. Here's someone with an occipital bun. And it's not obvious what the advantage of either of these would be, but they have persisted in the human population. I thought this was interesting, these life-size models of Neanderthal and modern human. Neanderthal, just to summarize, they came from Africa. They used tools, they had nuclear families, they buried their dead, they controlled fire. They had an average lifespan of about 30 years, and they interbred with humans 40 to 50,000 years ago. Any questions about this? If not, to bring you up to date with Neanderthal genes, here's something that was published earlier this month from the WebMD News. Certain Neanderthal genes are risk factors for developing COVID-19. There are several alleles in a region of chromosome 3. This would be human chromosome 3. linked to respiratory failure in severe cases of the disease. These alleles came from a Neanderthal who lived in southern Europe about 50,000 years ago. People with Neanderthal alleles on chromosome 3 have 3 times the risk of needing ventilation if they get the virus. And about 50% of people in South Asia and 16% of people in Europe carry the Neanderthal alleles on chromosome 3. Now, how do we know this? Svante Pääbo. He made this discovery, made this correlation. And here's a quote from him. "It is striking that the genetic heritage from Neanderthals has such tragic consequences during the current pandemic. This is something we are investigating as quickly as possible." So Neanderthal influences are with us all the time. Any questions or comments about anything I've said? This is the last slide. I have not put this slide set in your Canvas container yet, but I will. But first I want to add a summary slide at the end of it. And I will also construct sometime this week a worksheet with examples, types of questions that might be asked about evolution. And as I said earlier, I plan to have a quiz a week from today covering evolution and whatever we talk about on Monday, which at this point I don't know. Okay. That's it for today. Happy to entertain any questions or comments that you have about this or anything else about the course. Anything about quiz 6 that you have questions about for me or for Scott. And don't forget, Scott has a workshop starting a half hour from now.
It's looking forward to it.
Yeah, so show up if you can.
Thank you.
Okay. Thank you for coming. And I will see you next Monday with the next topic, whatever that might be.
Thanks, Sarah. You have a great day.
Fantastic.
Okay. Let's see, how do I get out of here?
Just hit the stop share. Some interesting stuff.
Yeah, evolution is great.
It is. And, just so much stuff we know already, and